we had discussed with the antigenic determinants in the immunoglobulin, and based on that, how you can classify the immunoglobulins. And in that, I had discussed with you the isotypic determinant that classifies the immunoglobulin into 5 different classes, where the antigenic determinants are present in the constant region domain of these 5 different classes of the antibodies, which identifies these constant region domains because they are of different protein nature. Say, for example, IgG is gamma protein, IgM is mu protein, like that IgA is alpha, D is delta, and E is epsilon. Like that, it is classified because of the difference in amino acid sequences of these constant region domains of the 5 different classes of the antibody. Now we will talk about today the allotypic determinants. What is meant by allotypic determinants? It is this determinants that subclassifies each immunoglobulin into subtypes. But there are only 2 classes of immunoglobulins which have subtypes. I don't know whether you remember, I had mentioned IgG1 and I had even given the structure of IgE class into 4 different subclasses: IgG1, G2, G3, and G4. So if you want to classify an immunoglobulin of the same family into subclasses, you have to identify the minor differences in that immunoglobulin. And what are these minor differences? It is found that in the allotypic determinants, once again, these minor differences are there in the amino acid sequences of the subclasses of immunoglobulin in the constant region domain only. The constant region domain of the heavy chain, the constant region domain of the light chain, both show few amino acid differences among these subclasses of the immunoglobulins. That is between G1, G2, G3, and G4. It is because of this difference in amino acids, when you inject these antibodies into other animal, the animal recognizes these antigenic determinants and produces the antibodies, and the antibody produced against the IgG1 will not be able to recognize the IgG2 because IgG2 has its own antigenic determinants which are different from IgG1. Based on this, the immunoglobulin G class has been classified into, even though 4 major subclasses, that is G1, G2, G3, G4, there exists about 25 different allotypic IgG molecules. In any human given person, there could exist about 25 different type of IgG molecules. Similarly, IgA also is classified into IgA1 and A2. But in A2, there are 3 subclasses. But it is mainly because of the difference in the kappa chain of the IgA molecule. So therefore, the allotypic determinants are only the differences in the kappa chain constant region of the IgA molecule. They are classified as kappa 1, kappa 2, and kappa 3. That is in the IgA2 subclass. In the IgA2 subclass, there are 3 allotypic determinants, but IgA1 doesn't have, but IgA1 and A2 are also allotypes. They can be differentiated by the constant region amino acid sequence. Now, how is this minor difference in the amino acid sequence makes a difference for the antigenic determinants? You just note down or try to understand. Let me spell out few amino acid sequence just arbitrarily. Say, for example, in the CH2 domain of the IgG1 class. Okay, let us say one sequence is there as asparagine, glutamic acid, leucine, threonine, and lysine. I repeat, asparagine, glutamic acid, leucine, threonine, and lysine. Another subclass of G1 molecule or G2 may have as amino acid sequence in the same region as glutamic acid, methionine, threonine, and lysine. If you had noted down, you would know that the first sequence that I had given you is asparagine, glutamic acid, leucine, threonine, and lysine. The second sequence that I gave you, which is the modification, glutamic acid, methionine, threonine, and lysine. The difference is between these two amino acids have been changed. Asparagine has been replaced by glutamic acid. Leucine has been replaced by methionine. Remaining three amino acids are same. This much of difference in the amino acid residues itself will be sufficient to bring about allotypic determinants. And that is how the one particular class of immunoglobulin can exist in different forms of allotypes. It is not going to make a much of a difference or problem for an individual, but sometimes it would be necessary when large volumes of antibodies or plasma is going to be given to a person for therapy. When immunoglobulin is there of a particular person which is going to be given passively to the another person, it is better to see whether there is any kind of differences in the allotypic determinants between the two individuals, that is the donor as well as the recipient, to avoid the precipitin complication reaction. Because the antibody, if it is developed because of the allotypic determinants, the person who receives the passive antibody will start developing antibody against the antibody that he has received and may start destroying it, and he will not respond to the therapy. That is the reason these allotypic determinants are very important if a person is to be given passive immunotherapy for any kind of disease. Okay. Now let us see what are idiotypic determinants. Idiotypic determinants are also called as idiotopes in the similar manner as we call antigenic determinants as epitopes. Epitopes are nothing but antigenic determinants. Now, idiotypes are purely based on idiotopes situated in the FAB domain of the antibody. Previous two classifications that we spoke about, that is isotypic determinants, is based on the antigenic determinants of the constant region domain of the heavy chain and light chain. Similarly, allotypic determinants is also antigenic determinants of the constant region domain of the heavy chain and the light chain. Whereas in the case of idiotypic determinants, it is the determinants that are situated in the FAB domain, FAB region of the antibody. The FAB region of the antibody is supposed to be containing variable region of the heavy chain and variable region of the light chain. And in that, I had also classified that into three zones of hypervariable regions, which are also called as complementarity determining regions. Remember, they determine the specificity of a particular antibody with regards to the antigenic epitopes or antigenic determinants. So, For a given antibody of a particular class, let us say I had told you that IgG is a predominant antibody produced in large quantities against any antigen. And I had said that a particular pathogen can have more than one antigenic determiner. Let us go back to the same example of Salmonella. In Salmonella, I had mentioned about three major antigens of the membrane. One of them will be flagellin, another will be the antigen O, that is of the membrane antigen, and another is antigen H. So these three different antigens of the Salmonella will be recognized as three different epitopes, and three different IgG molecules will be produced by three clones of B lymphocytes. Three different B lymphocytes produce three different IgG molecules, but they belong to the same class. And these three IgG molecules differ from one another even though they belong to the same allotype. See here in the example they have shown mouse IgG1, IgG1. Even though they belong to the same allotype, they differ in their FAB site because each FAB site of that antibody is meant for one particular antigenic epitope. If one IgG1 molecule is meant for H antigen, another is meant for O antigen, and another may be meant for flagellin. So based on these differences, because of the amino acid sequence difference in the FAB region, they are different in their FAB site because those amino acid sequences are meant to be interacting with the antigenic epitopes by the interaction which we had mentioned in my first lecture, that is through weak van der Waals forces or ionic bond or hydrogen bond or hydrophobic interaction. And that affinity is based on that FAB region on that antigenic epitope. So therefore, that antigenic epitope is an idiotope, which differs from one class of antibody, that is IgG, to another IgG molecule because it is highly specific as far as the antigenic determinants or epitopes of the antigen is concerned. And therefore, these idiotopes or idiotypes play a major role in the sense in autoimmune response, you know that the individual suffers from a disease where his own antigens are going to be attacked by his own antibodies. And those antibodies are produced against autologous antigens, that is, self antigens. And these antibodies destroy these antigens and cause tissue damage and cause damage to the organs. We'll be talking about that in detail when we are going to talk about autoimmune response in Unit 4. But for the time being, you keep in mind the antibody, self antibody, that is autoimmune antibody produced against the self antigens will also contain idiotopes or they are idiotypic. If you want to control or develop a therapeutic measure against the autoimmune response, you can design a monoclonal antibody against the idiotope of the autoimmune antibody. The autoimmune antibody has idiotopes. Now you have to develop a monoclonal antibody against those idiotopes of the auto antibody. And those antibodies developed against the idiotopes are called as anti-idiotypic antibodies. And these anti-idiotypic monoclonal antibodies are used to destroy the autoimmune antibodies. And they are given as a therapeutic measure to the individuals who suffer from autoimmune disorders of different types. This is how you can save the person from the autoimmune disorder by developing anti-idiotypic monoclonal antibodies. And those monoclonal antibodies come and bind to those idiotopes and prevent that autoimmune antibody from binding to the self antigen, and thereby it will prevent the destruction of the self antigen or the organ. You know that when the antibody forms a complex with the antigen, the complement mediated lysis reaction will start. That is how, when the autoantibodies bind to the self antigens, they may destroy the organs by complement mediated lysis. To prevent that, you have to prevent the autoantibody from binding to the self antigen. And how you do it? By synthesizing or by creating the monoclonal antibody against the idiotopes of the self antibody and prevent that from binding to the self antigen. That is as far as idiotypic determinants are concerned. So, for the time being, we have finished with the topic on types of antibodies. Now, we will talk about the organization of the immunoglobulin gene. You will understand the immunoglobulin genes which are arranged in the chromosome of an individual, how the constant region and how the variable region domains of the antibody are brought together to construct an antibody molecule. Now I would like to know whether you can see the slide. I have changed the slide. The antibody genes in the human beings, I had mentioned that they are situated on different chromosomes. Say for example, the lambda chain light chain gene is present on chromosome number 22. The kappa chain light chain gene is present on chromosome number 2. And the heavy chain gene is present on chromosome number 14. Okay, first I will take the organization of the gene on the kappa chain of the chromosome number 2 because it is the simplest. It is the simplest. We kept on mentioning the antibodies have variable region, and the variable region constitute the FAB site. Antibodies have constant region, and the constant region constitutes the part of the light chain constant region, and the constant region of the heavy chain constitute the part of the heavy chain. The variable region has to recombine, means it has to be brought closer to the constant region, and it is the only specialized incidence of recombination within the chromosome in the somatic cell that is observed in antibody producing B lymphocyte. No other somatic cell in our body shows this type of recombination, that is rearrangement of the genes, means the variable region segment is to be combined with the constant region segment. Now let me take in the kappa chain. In the human being, the variable region genes are about 300. The N85 shown here is for the mouse. Same arrangement is there in the mouse, but the chromosome will be different, and number of variable genes in mouse are about 85. That is not very important for you to know. Okay, but for the human beings, about 300 variable region genes are present. And these 300 variable region genes are spread out into a large segment, into a cluster. Here it is shown as VK1, VK2, VKN. So means each gene segment, that is an exon that codes, is accompanied by a leader sequence, that is L. The leader sequence is always on the 5' side of the exon. each exon is accompanied by a leader sequence. And that leader sequence directs the variable region domain to combine with the constant region domain in that same order in the 5' to 3' direction. It determines the recombination order in the 5' to 3' direction. How that recombination takes place, we will talk about it little later. So 300 clusters of the exons of the variable region of the kappa chain distributed on the chromosome number 2 in the upstream region of the constant region. Upstream means towards the 5' end of the constant region. Now, in between the constant region gene and the variable region gene, there is a gene segment once again called as J. J is for joining or junctional segment, joining segment or junctional segment. There are in the kappa chain 5 J genes, J1, J2, J3, J4, J5. Joining segments, 5. And these joining segments are also separated by introns. And each segment of the J will code for about 13 amino acids, 13 amino acids. Of the 5 J segments in the kappa chain, one of the segment is non-functional, that is indicated by the psi symbol, psi, the Greek symbol, psi symbol. And that segment does not play any role, and it is not known why it is still existing. It is retained there, probably to maintain the distances between the exons of the genes. You know that there are junk DNAs present in the intronic regions and in between the exons, and those junk DNA segments keep the genes apart, and it is necessary to keep the genes apart to facilitate recombination. That may be the reason probably the J size segment is existing between the two functional J segments. Now that J segment is present closer to the constant region domain. In the kappa chain, there is only one constant region domain. That means if the kappa chain or kappa gene is chosen, and I have told you that majority of the immunoglobulins, about 60% of the immunoglobulins that are produced in our body choose kappa chains. It may be because it has got only one constant region that has to recombine with any 300 variable region genes to produce a productive gene product with FAB site having high affinity for the antigen. Okay, high affinity. And that variable region gene that is chosen should have the antigenic determinants to bind to the antigens. Okay, productive with high affinity or low affinity. And the one segment that does not show any affinity, that is useless. Now, how does this chosen is because of the J segments of the gene present between the constant and variable facilitate the fusion of variable region gene exon with the constant region gene by having one of the J segment in between them. So therefore, in the variable region of the antibody, 13 amino acids of the 3' region. This is the 3' region of the variable region, and this is the 5' region of the variable region. So 3' segment of the variable region, 13 amino acids that are included in the variable region belong to the J segment of the genes. Those 13 amino acids will become the part and parcel of the variable region when it recombines with the constant region. How it recombines, we will see little later. Then we will go to another light chain, that is lambda chain. The lambda chain, unlike the kappa chain, is slightly complicated. It is present on the chromosome number 22. Okay, there are about 6 constant region genes present. All are not shown here. 6 constant region genes are present. They are spread out into a large segment of the chromosome number 22. Okay, and then probably one or two constant regions here in the diagram, two constant regions, that is C lambda 2 and C lambda 4. Okay, C lambda 2 and C lambda 4 of the 3' end are shown with one variable region gene. Okay, one variable region gene on the 5' end. Similarly here, C lambda 3 and C lambda 1, that is constant region domains C lambda 3, C lambda 1, are shown with V lambda 1 on the 5' end. It doesn't mean that if there are 6 constant region genes, there are only 6 variable region genes. No. On the 5' end of each constant region clusters, this one segment with a break in between shown here to indicate that that is not the end of the chromosome. It is continuing like that with different gene clusters. Each constant region gene cluster is accompanied by large number of variable region genes on the 5' end. Even though it is shown as V lambda 2 here, there could be other variable region genes on the other side of the 5' region. So each constant region gene has its own cluster of variable region genes. Here also once again about 300 variable region genes are present in the 5' end of the constant region gene cluster. But each constant region gene is accompanied, there are only 6. Already 4 are shown here. There should be one more segment with 2 more constant region genes, but clusters of variable region genes are many with each constant region cluster. But each constant region gene in its 5' region upstream accompanied by one J segment. If there are 6 constant region genes for the lambda chain, there are 6 J segments. Each constant region is accompanied by 6 J segments. And those 6 J segments are accordingly named after the constant region. So for example, if the constant region is C alpha 2, the J segment will be J alpha 2. Similarly, constant region segment is C alpha 3, the J segment will be J alpha 3. And it is always on the 5' end of the constant region, but separated by an intron. There is a gap. Similarly, there is a large gap between the J segment and the variable region gene. Similar to the leader sequence that is present in the 5' end, which we had seen in kappa chain, here also the variable region gene is accompanied by a leader sequence in the 5' end. Each variable region gene has its own leader sequence. That is to indicate the recombination should take, it is a regulatory sequence. The recombination should take place in the 5' to 3' end. That variable region of the 5' region should recombine with the constant region lying at the 3' end using the J segment in between as a junction or joining segment. So 6 clusters of constant region genes accompanied by 6 J segments and large number of variable gene clusters are present in the 5' end of each constant region clusters, and that constitute the lambda chain of the light chain. So it is slightly complicated than the kappa chain. Therefore, the recombination here will be between the each cluster. The variable region, let us say, of V alpha 2 recombines. It has a possibility of recombination with the C alpha 2, or it has the possibility of recombination with the C alpha 4. Similarly, variable region V alpha 1 has the possibility of recombination with C alpha 3. At the same time, it has the equal opportunity of possibility with C alpha 1. Like that, other two clusters, C alpha 5 and C alpha 6, will be there with variable region gene. They also have independent ability to recombine. So whichever the segment of the light chain of the lambda chain is chosen, it should produce a productive gene product with the high affinity epitope or high affinity FAB site or low affinity FAB site. Otherwise, that gene product will be useless. So let us see now the heavy chain gene that is present on chromosome number 14. It is a large segment because 5 different classes of the immunoglobulins are produced from here. See the constant region domains that are present in the 3' end of the genetic segment, which is spread out into different segments with introns in between. The first two genes of the constant region are always C mu. Next to it is C delta. The constant region for IgM, constant region for IgD. Why? Because you know that the first receptor on the B lymphocyte for the recognition of the antigen, that is pro B cell. Okay, sorry, no, pre B cell. From pro B cell, when the pre B cell is produced, the gene that is chosen for putting an antigenic receptor, which is MIgM molecule, that is always a M molecule, IgM. Therefore, this gene is the first gene, which is the first gene to be chosen by the variable region domain to recombine because it is nearest to the variable region gene. Other genes are far away. Okay, so C mu and C delta constitute the receptor of the B lymphocyte, which I had discussed yesterday also, and when I had discussed with the B cell receptor. So therefore, these two genes are the first two genes of the constant region that are present in the 5' end of the genetic segment to recombine with the variable region gene. Then comes the 4 different classes of the gamma globulin, and those gamma globulin genes are present here immediately after the C delta. Then comes the C epsilon, and the last region at the 3' end is C alpha, that is IgA. Okay, so IgM first, IgD second gene, third is Ig gamma, that is IgG, and fourth is Ig epsilon, and the fifth is IgA or alpha. Here in the human beings, on chromosome number 14, the gene cluster is distributed in a large segment in such a way that there is additional segment between the variable region gene and the constant region gene. Here in the case of light chain, we had spoken about variable region gene and constant region gene. In between them, a J segment is present. But here in the case of heavy chain, in addition to the J segment, there is D segment. D is for diversity. T is for diversity, J is for joining, V is for variable, C for constant. So the recombination in the heavy chain follows the order of V, D, J, C. That is, variable region domain first recombines with D, that is diversity, then it recombines with J, then it recombines with mu or delta or gamma or epsilon, like that. So therefore, recombination in the heavy chain will be VDJC. Recombination in the light chain will be VJC, VJC, J for joining. So in the case of heavy chain, the joining segments are also about 4 to 6 J segments, or 4 to 6 in the human beings. Here what is shown is of the mouse, and the diversity segment, what is shown here is about 13, but in the human beings there are about 30, 30 diversity segment. The diversity segment combining with joining segment together gives rise to large amount of variation in the variable region, that is going to be in the 3' end of the variable region. It forms the exonic component and the coding region and constitutes the amino acids of the variable region domain of the heavy chain. So D and J constitute some amino acids. 13 amino acids come from J segment. Another 13 or so come from the D segment. 13 plus 13, about 26 amino acids. They will be contributing to the variability of the variable region gene. Similar to the leader sequence that was seen in the light chain, here also 300 variable region genes are going to be accompanied by a leader sequence in the 5' end. Okay, leader sequence. Now, any of those variable region genes that are chosen, which have to combine first with D, then with J, VDJ recombination, then with the C region. So the recombination that takes place in the 5' to 3' order in the VDJC recombination will choose the constant region domain. But always it is the thumb rule. M, that is C mu gene, cannot be skipped. It has to be chosen as the first gene to produce the antigenic receptor on the B lymphocyte. Without choosing C mu gene, antibodies are not going to be produced. Then the second gene that has to be chosen is C delta. Therefore, these two genes are placed very close to each other, and they are much towards the 5' end, and they're closer to the variable region domain, so that they are going to be chosen first genes to put the B cell receptor. And whenever that choosing of the gene takes place, it is random. And the C mu, which is spread out, even though they have shown one block here, that is further into a large segment running into thousands of nitrogenous bases. Okay, thousands of nitrogenous bases. And within that, there are some more introns present. And those 1000 nitrogenous bases, which are going to be chosen as the segment of the C mu gene, when it recombines with any of the variable region gene among the 300, if it produces a high affinity or low affinity receptor with the FAB side, then only that gene is going to be chosen and used for the development, further development of the antibodies. Then first produced antibody will be always the IgM, and IgD very rarely going to be put into circulation. I have mentioned it to you already that it is about 0.03 milligram per ml. But IgM, first 5 days of the individual's immune response comes from the IgM. Then class switching takes place. That means the gene is going to skip the same variable region that was chosen to recombine with the C mu to produce a productive receptor of high affinity or low affinity. The same variable region now is going to jump and choose any one of these gamma genes, that is of the IgG class or epsilon class or alpha class, and that jumping is called as class switching. First produced antibody is IgM, then the class switching should take place and it should start producing large quantities of IgG because it will be in circulation to defend and act against the antigen. So this process of recombination between the light chains, between the heavy chains, we will discuss that in the next lecture. Thank you very much.